Now let's take a look at how the auditory system allows hearing to happen. And it actually begins with the outer ear. So you can see here the pinna. That's just your outer ear. The pinna is actually important because it helps to amplify the frequencies between 2,000 and 5,000 just about. So it's very handy for amplifying the frequencies that you would encounter human speech in. We also have here the auditory canal, the external auditory canal, and then so the sound waves, imagine them coming on in until they hit the eardrum, also known as the tympanic membrane. That now transitions us to the middle ear. And notice these three bones here. They are the hammer, the anvil, and the stirrup. Those three bones work like a set of levers to amplify sounds as well. And we can refer to them collectively as the ossicles. And the last little bone here, the stirrup, taps onto the round window of the cochlea, which we see here kind of coiled up. Inside the cochlea, in a fluid, are these hair cells. And so when the stirrup taps on this area, it makes these hair cells bend, and that leads to the neural signal in the form of increased firing or decreased firing, depending on the direction that the hair cells are bending. So they tend to bend first one way and then another way as the sound wave goes past in the form of this fluid that's in here inside of the cochlea. The Eustachian tube, you may have heard about that. That equalizes pressure of the middle ear with the pressure of the environment. And also here in the middle ear, we have the tensor tympani. That's a muscle that adjusts the tension of the eardrum, and it helps to protect us a little bit from loud sound. Here's a closer look at the ossicles. So we can see here the stirrup, the last one, and the anvil right here, and the hammer. And so the three work like a series of levers to amplify the small vibrations that are received here at the tympanic membrane and make them larger vibrations that then travel through the cochlea. Note that the Latin names of the bones of the middle ear are malleus, incus, and stapes. So hammer in Latin is malleus, anvil in Latin is incus, and the stirrup is referred to as the stapes. Don't worry, we will be using the English terms for the class. The inner ear includes this oval window into the cochlea. And we have in the cochlea if you look down here on the bottom right, a basilar membrane and a tectorial membrane, kind of like the two slices of bread on a sandwich. In the middle are the hair cells. And they're cells, and you can see their cell bodies and so on. The hair cells have little cilia on the ends, and so they act kind of like seaweed does, and they will flow and bend right and left. And as they do so, they release potassium and calcium in sync with those vibrations. And also, depending on the direction that they move, remember, you'll get an increase in firing or a decrease in firing. So you get this pattern of increased firing and then decreased firing as the sound wave goes through. And then the auditory nerve, which is the eighth cranial nerve, will send this information from the hair cells to the brain. Here's a closer look of where the hair cells are. And notice that there are outer hair cells and inner hair cells. The inner hair cells are the receptors for hearing. So these guys are the ones that are creating that neural signal. They are the ones that are responsible for sensation. The outer hair cells play an important role, though. They are involved in amplification and a sharpening of the sound. In other words, the ability to distinguish between similar frequencies.
Okay, so now let's take a closer look at the cochlea and the inner ear. Let me just draw a little cochlea. So it's this round snail-like structure. But let's go ahead and unroll it. So let's just unroll it and lie it flat. So if we unroll it, it'll look something like this. It'll look something like that. So basically this is just a flattened cochlea. And so you've got this little bone that we were talking about earlier, which is called the stapes. So you've got this little bone right here. It's called the stapes, and this stapes is connected to the other two bones and then the eardrum. So it's basically moving back and forth at the same frequency as the sound wave that's making the eardrum move back and forth. And this stapes is connected to this little oval membrane called the elliptical window. And the elliptical window gets pushed in and out as the stapes moves back and forth. So as the elliptical window gets pushed in, there's fluid inside the cochlea. So we've got fluid inside, and that fluid basically gets pushed all the way around the cochlea and then comes back around. And as I mentioned before, the reason that the fluid flows in this direction is because there's actually a structure right in the middle. There's this little structure, and we call it the organ of Corti. So this organ of Corti kind of splits the cochlea in two, and the fluid can only flow in this direction. So when the fluid gets over here, there's this little round window, it's actually called the circular window, and the circular window gets pushed out a little bit as the fluid kind of compresses it. So now you've got fluid flow back and forth around this organ of Corti. So what we want to look at now is we just want to look at a cross-section, just a little cross-section of this organ of Corti, so that we can kind of understand what happens. How does it turn this fluid motion into sound? So if you actually kind of look at a cross-section, what you would see is something like this. So something like this. So you've got an upper membrane and a bottom and a lower membrane, and you've also got these little hair cells. So you've got little hair cells with little shark fin looking things on the tops. So you've got these little hair cells, and basically as there's fluid flow around this organ of Corti, it goes like that, and then the fluid kind of comes around and goes this way. So as we have fluid flow, it actually pushes down on this membrane and pushes up on this membrane. So you can kind of imagine how this fluid flow works. So as this membrane gets pushed up and down, it actually causes these little hair cells to move back and forth. So they're moving back and forth, they're vibrating, and basically what we can do is we can blow up these hair cells. So we can blow them up and look at them in a little bit more detail. So it kind of looks something like this. So there's their shark fin part, and then there's the cell. So this shark fin part actually is called the hair bundle. Hair bundle. And these aren't actually hairs. What they are is they're just a bunch of little filaments, and let me just draw it in a little bit bigger. So if we were to look at just the hair bundle, it would look something like this. It would look something like this. And each one of these filaments is called kinocilium. Kinocilium. So basically, a whole bunch of these little filaments are attached to one another, and they form the hair bundle. So each kinocilium is actually connected to one another by this little spring-like structure called a tip link. So it's a little spring-like structure, and each one is called a tip link. So it links the tips of the kinocilium. So if we were to actually look at a tip link, so let's go ahead and look at just the tip of this kinocilium. If we were to look at just the tip of this kinocilium, it would look something like that. And you've got the little tip link attached to it. So you've got this little spring-like structure attached to the tip of this kinocilium. And in fact, the tip link isn't attached to the kinocilium directly, but it's actually attached to the gate of a potassium channel. So there's the little gate right here. So this is the little gate of a potassium channel. And so as these hair cells, as the little kinocilii get pushed back and forth because the fluid is moving in the cochlea, as they get pushed back and forth, it actually stretches on this spring. So let's say that the kinocilium gets stretched. Sorry, we're going to use this color over here. Let's say that the kinocilium gets stretched. It actually kind of looks like this. So now it's getting stretched, and as it gets stretched, it actually opens up this gate. So as the potassium channel gate opens up, there's potassium outside that then flows into the cell. So you've basically got potassium out here flowing into the cell. And there are actually all these other little channels, calcium channels, that get activated when potassium is inside the cell. So now you also have calcium flowing into the cell. So the flowing of potassium and calcium into the cell basically causes the cell to fire an action potential. So it basically stimulates another cell, which is known as a spiral ganglion cell. And the spiral ganglion cell then activates another cell that is part of the auditory nerve, which then goes to the brain. So basically this goes to the brain. So this is what happens when a sound wave comes into the ear and then gets transmitted into a neural impulse by these little hair cells.
And again, the auditory nerve, or also known as the eighth cranial nerve, sends this information up to the brain. And so we can see here the auditory nerve is going to first stop at an area called the cochlear nucleus. That's aptly named because it's receiving information from the cochlea, right? From the cochlea, it goes up to this area in the midbrain called the superior olives. The superior olives, besides having a really cool name, are important because they help us locate the source of a sound. From the superior olives, the auditory signal travels up to this area called the inferior colliculus. Right here, this is in the midbrain. And the plural, because there's one on the left, one on the right, is the inferior colliculi. So there are two of them. They bring together the sound from both ears, and they create an important auditory map of the environment around us so that we can locate the source of a sound easily in our environment. And it also will immediately produce a reflex to a sound that you weren't expecting. So a sound on your right that you weren't expecting might get you to turn your head to face that direction. And that is a reflex that the inferior colliculus does. From the inferior colliculus, the information goes up then to the thalamus. So now we're in the forebrain. And remember, in the thalamus, the part of the thalamus that receives auditory information is the medial geniculate nucleus. So remember M for music, that'll help you associate it with sound. And the medial geniculate nucleus, also abbreviated MGN, receives mostly information from the opposite ear. And then from there, it goes to the primary auditory cortex here in the temporal lobe.